So we have some enzymes that are working to build glycogen and other enzymes that are working to tear glycogen down. And so you can imagine we definitely need to control those enzymes. And in control of glycogen metabolism is an important thing to consider. So there are two ways we're going to look at this. One is allosteric controls, and that's something we've visited before, thinking about phosphofructokinase and how ATP and AMP modulate PFK's activity. In glycogen metabolism, we're going to look at two enzymes and focus on them intently, and that is the phosphorylase and the synthase. So remember, glycogenolysis, the process of breaking down glycogen, involves the enzyme glycogen phosphorylase. So let's think about glycogen phosphorylase in terms of how it is activated or inhibited by other things. So it is activated by increasing levels of AMP in the cell. And that should make sense because glycogen phosphorylase breaks down glycogen, and glycogen was, of course, a storage form of glucose. So now that glucose can run through glycolysis and produce energy in the cell. So high AMP levels indicated lower energy state of the cell. The cell needs to make ATP. Let's break down glycogen. And then it might make sense that it is inhibited by ATP, right? So if energy levels in the cell are high, so we've got a lot of ATP, well, we don't need to break down more glycogen. But it's also inhibited by glucose 6-phosphate, because if we have a lot of glucose entering the cell from the bloodstream, say after a meal or something like that, we don't need to break down glycogen because we apparently have another source of glucose. The other enzyme, of course, we have to think about in glycogen synthesis is glycogen synthase. And the one I want to concentrate here mostly is this, that it is activated by glucose 6-phosphate. And the way to think about that is, again, if there's a surplus of glucose, say in the bloodstream entering the cell, it's going to produce glucose 6-phosphate. Let's store that away as glycogen. Also, if glycolysis activity has decreased because the cell doesn't need energy, then glucose 6-phosphate levels would increase, and we need to store those away. So those are some allosteric controls of glycogen metabolism, and I'll point it out again. Concentrate when we're talking about glycogen breakdown. The phosphorylase is the most important enzyme, and when we're talking about glycogen production or synthesis, the synthase is the most important enzyme. The other enzymes we've talked about matter, but we're gonna focus most intently on these two. So one other way that enzymes can be modulated is through covalent modification. And in general, this means we're going to add some group. Could be an acetyl group, a methyl group, phosphate, could be other things. It's sort of a post-translational modification, if you want to think of it like that. And these change the protein's conformation. Maybe they block a binding site or an active site or something like that, or put the protein into a productive conformation. And we're going to focus in on phosphorylation here in the case of glycogen metabolism, and it's usually a serine, threonine, or tyrosine group that's phosphorylated. So one of those OH containing amino acid side chains. And of course, we already know that kinase enzymes use ATP to add a phosphate group to something, and that's indeed the case here. So anytime we're adding a phosphate, we're going to use a kinase enzyme. And just like we saw before, anytime we are removing a phosphate group, we're going to use a phosphatase enzyme that's using water in a hydrolysis reaction to remove that. So we're going to talk about kinases and phosphatases and the way they modify glycogen phosphorylase and glycogen synthase. And so these two enzymes are subject to phosphorylation, as I mentioned, and we need to identify and define forms of covalently modified enzymes. And so we're going to say that these enzymes in general can exist in two forms. There's a form A, or an A form, that is the active form, and a less active or inactive form, that's B. So A is active, B is inactive or barely active, if you want to have a mnemonic there. So there's a key phrase here, is that phosphorylation activates glycogen phosphorylase. That is, adding a phosphate group to glycogen phosphorylase converts it from the inactive B form to the active A form. And then conversely, phosphorylation deactivates glycogen synthase, so converts it from the active A form to the inactive B form. And so there are several enzymes involved here, and I'm not going to talk about these on this slide. There's on the next slide we're going to get into how the whole system works in a little more detail. But there's a key phrase that I want you to remember, and that is that phosphorylation activates the phosphorylase. Phosphorylation activates the phosphorylase. If we remember this, then a lot of the way glycogen metabolism operates is going to fall out of that key phrase. So this might look intimidating, and it is intimidating. We're going to simplify things down a little bit here, and that is that we're going to focus first on the inner part here. I don't want to make this too messy, but notice what we're looking at. We're looking at our two key enzymes, and that is glycogen phosphorylase and glycogen synthase. And as we have just said, that phosphorylation activates the phosphorylase. So if glycogen phosphorylase is subject to a kinase activity called phosphorylase kinase that uses ATP to add a phosphate group to it, then glycogen phosphorylase becomes active. And conversely, if glycogen synthase is acted upon by a kinase, phosphorylating it then converts it to the inactive form. And then removing that phosphate activates the synthase, and removing the phosphate group from the phosphorylase deactivates that enzyme. And so that's sort of the key part here, is that there are kinases that are acting to add phosphate groups, and there are phosphatases that are acting to remove phosphate groups. And then the removal or addition of phosphate groups has opposite effects on the phosphorylase versus the synthase. And so we have a phosphorylation system up here whose job it is to add phosphate groups, and we have a dephosphorylation system whose job it is to remove those phosphate groups. And so let's just think about how this goes about. Let's say, for example, that we have turned on the phosphorylation system. That means that glycogen phosphorylase is going to be switched to the on, and glycogen synthase is going to be switched off. And so the phosphorylation system, if you think about it, it's going to favor glycogenolysis, right? We are turning on the glycogen phosphorylase, which breaks down glycogen. We're turning off glycogen synthase, which of course builds glycogen. So anytime the phosphorylation system is activated, it means that glycogenolysis is going to occur. And it also means that the cell probably needs energy. And that's why that system has been turned on. I should say favors glycogenolysis here. And then we have the dephosphorylation system, which is going to then remove phosphate groups, as I said. And so that will turn off glycogen phosphorylase and turn on glycogen synthase. So the dephosphorylation system favors glycogen synthesis. And the dephosphorylation system is probably turned on because the cell has an abundance of energy and does not need ATP. So we're going to stop breaking down glycogen, and instead we're going to store glycogen away. So I don't care that you spend as much time with these details within the system, you can sort of more or less treat the dephosphorylation system and the phosphorylation system as black boxes. I care more that you focus in on the inner part here and the effect that the dephosphorylation or phosphorylation system has on those. And so I just want to briefly talk about hormonal control of this whole system and how glycogen is controlled by hormones for sure. And this one right here is insulin. It is a protein that is manufactured in the liver. And then we have glucagon here. Glucagon and insulin operate to control blood sugar levels, as you probably know. So insulin is this folded up protein that's been processed, and glucagon is just this little teeny alpha helical protein shown there. So I want to explore how these things operate within in sort of an organismal level, thinking about humans here. And so we're going to explore what happens with a muscle cell and also with a liver cell and how these things operate differently. And remember, I have said that muscle cells are selfish and liver cells are selfless, and we're going to see how that operates. So recall that the pancreas actually is critically important in blood sugar control. And let's say after a meal you have a high amount of blood sugar, and the pancreas detects that and responds by releasing insulin. Insulin is a key marker that their blood sugar levels are high and cells need to take up blood sugar, glucose, and do something with it. In the muscle cell, there are insulin receptors that will identify that insulin is in the blood, and it will do a few things. It's going to activate these glucose transporters. It's actually going to produce more of these glucose transporters to enable the glucose that's within the blood to enter the cell. And that insulin will also trigger glycogen synthesis. It's going to turn on the synthesis of glycogen to store away that glucose in the form of glycogen. Some similar things happen in the liver. Insulin is a signifier to the liver to also synthesize glycogen, store that away by taking in glucose from the bloodstream. So insulin responds when blood glucose levels are high. Let's look at what happens with this other hormone, glucagon. And the way I remember this is the pancreas releases glucagon when glucose is gone, or at least at low levels. This has nothing to do with muscle cells. Notice muscle cells are not involved in this process because they're selfish. They don't care if the blood glucose levels are low. All they do is take. The liver, on the other hand, will recognize that there is glucagon present, and it will enable then the glycogen degradation pathway to be activated, shown here, and that's going to produce glucose, and that glucose can be delivered out into the bloodstream or exported out into the bloodstream. So the liver responds to glucagon by releasing blood glucose, or I should say glucose, into the blood so that those levels increase. And I also want to look at epinephrine. So epinephrine is a hormone as well, and it has similar effects in both tissues. Remember, epinephrine or adrenaline is the fight or flight hormone, and so it is what we release in order to run away from something or get the energy to do something dramatic. And so there are these adrenoreceptors on the surface of muscle cells. They recognize epinephrine, and they then go through and trigger glycogen degradation, which then provides energy for glycolysis, and so our muscles can run away from some dangerous thing. It's also recognized on the surface of liver cells, and it will do the same thing where it enables glycogen degradation and exports more glucose to the bloodstream so that glucose can essentially be taken up by muscle cells and the brain as well in those stressful situations. So what we should know from all of this is know the roles of insulin, of glucagon, and of epinephrine, and how those hormones influence muscle cells and liver cells differently.